This is the cell cycle. DNA replication occurs in the middle of the cell cycle during the S phase, also known as DNA synthesis.
In this picture, you can see an enzyme called the DNA helicase. Helicase unzips the DNA by breaking the hydrogen bonds between nitrogenous bases. Helicase breaks specifically the bonds of adenine, thymine, cytosine, and guanine, which forms a replication fork. As you can see, the two strands lagging and leading are fully separated and ready for the next step.
Now, a new enzyme gets added after the helicase successfully breaks apart the original parent strand. The lagging strand on the right is lagging since it moves from the strand's 3' end to the 5' end. The leading strand moves from the 5' end to the 3' end, making it continuous for DNA polymerase. The new enzyme is primase. Primase goes in and creates a starting point called RNA primer for the new strand to be created.
After primase attached RNA primers to each of the original template strands, DNA polymerase comes in. As you can see in the video, DNA polymerase is working in the anti-parallel pattern. This enzyme is responsible for attaching new nucleotides to eventually form a complementary strand on both strands of the DNA template. Unlike the continuous formation of the leading strand, the lagging strand is formed in chunks. These chunks are called Okazaki fragments.
Once the DNA polymerase has gone in and attached the nucleotides, an enzyme called ligase comes through and makes sure the lagging strand's Okazaki fragments are connected. Ligase is the glue for the lagging strands so they can successfully stick together as one continuous complementary strand.
In the end, DNA replication made 2 new completed deoxyribose nucleic acids.